Welcome you all for the cancer introduction part 3. Here I'll discuss about the involvement of genetics into the cancer. Approximately 90 to 95% of all cancers are sporadic in nature, while 5 to 10% are inherited. So genetical involvement is 5 to 10%. Our cells are genetically programmed to have particular lifespan and to divide or die. One gene is known to act as a brake on cell division, another gene enables cells to live indefinitely and keep on dividing. Any imbalance in this activity of these genes may lead to the abnormal cell division. Often the malignant cells are carried by the lymph or blood or other organs such as the liver where secondary tumors develop. So what is happening here? There are two kind of systems working, where one type of system which is promoting the cells to divide and the type of system which are inhibiting cells to divide. So whenever an imbalance created between these two type of systems, there is a cancer, there is a chances of cancer development. Cancer is caused in all or almost all instances by mutation or by some other abnormal activation of cellular genes that control cell growth and cell mitosis. The abnormal genes are called oncogenes. As many as 100 different oncogenes have been discovered. Also present in all cells are anti-oncogenes, which suppresses the activation of specific oncogenes. It means what? There are two type of genes which are supporting these two systems. There are oncogenes and anti-oncogenes. Oncogenes which are supporting the cell growth and proliferation or cell development or cell division, where the anti-oncogenes suppressing this system. So whenever there is an imbalance between the oncogene or anti-oncogene, the cancer development chances increases. Therefore, loss of or inactivation of an anti-oncogene can allow activation of oncogenes that lead to cancer. Only a minute fraction of the cells that mutate in the body ever lead to cancer. There are several reasons for this. First, most mutated cells have less survival capability than normal cells and simply die. Second, only a few of the mutated cells that do survive become cancerous. So these are the two conditions here. One, highly most mutated cells has less survival capability, and second, the few of the mutated cells that do survive become cancerous. Okay, so only those mutated cells which are having survival can convert into the cancerous cells. Third condition, potentially cancerous cells are often, if not usually, destroyed by the body's immune system before they grow into a cancer. People taking immunosuppressant drugs after kidney transplant or something can be more dangerous for that. Fourth condition, usually several different activated oncogenes are required simultaneously to cause a cancer. For instance, one such gene might promote rapid reproduction of a cell line, but no cancer occurs because there is not a simultaneous mutant gene to form the needed blood vessels. What does it mean? This is a very intricate and complex system, even for causing the cancer. That's why cancer is one of the rarest disease. So for developing a cancer, even you need a various genes together simultaneously to work with. Many trillions of new cells are formed each year in humans. Why not all of us develop millions or billions of mutant cancer cells? The answer is the incredible precision with which DNA replication takes place in each cell before mitosis, and also the proofreading process that cuts and repairs any abnormal DNA strand before the mitotic process is allowed to proceed. I think you all remember the protein synthesis and RNA splicing process by which a different kind of genetic material has been produced and that different kind of cells has been passed. So that precision of RNA splicing or the DNA splicing, that actually gives you the different variations. Yet despite all these inherited cellular precautions, probably one newly formed cell in every few millions still has significant mutant characteristics and forms a large number of cancer cells. The probability of mutation can be increased manifold when a person is exposed to the following factors. Number one, ionizing radiation. For example, X-rays, gamma rays, and particle radiation from radioactive substances, and even ultraviolet light can predispose individuals to the cancer. So you remember always the ionizing radiations are one of the causative reason for cancers. Chemical substances, or we call it as carcinogens, as aniline derivatives. The carcinogens that currently cause the 1/4 of all cancer deaths are those in cigarette smoke. Next is the physical irritants, such as continued abrasion of the linings of the interstitial tract by some type of blood, some type of food. The damage to the tissue leads to rapid mitotic replacement of the cells. The more rapid the mitosis, the greater the chance of mutation. So here by physical irritants, there are some kind of chemical which physically affect the lining of the cells and make them mutant. Hereditary tendency, certain type of viruses can cause some kind of cancer including leukemia. DNA and RNA viruses causes mutations generally in lab animals. So here there are other reasons like RNA and DNA viruses can also do that. Oncogenic RNA viruses all appear to contain a reverse transcriptase enzyme that permits translation of the RNA message of the tumor virus into the DNA code of the infected cells. A specific human retrovirus have been identified as being causative agent for a specific type of human T cell leukemia. So HTLV1 is also a kind of virus which causes the human T cell leukemia, which is another very serious kind of cancer which directly affects the immune system of the human being. These oncogenic RNA viruses are causing very diverse kind of cancers, and later in the slides you will understand these different kind of cancers here. Thank you.